An additional consequence of the nonspecific activation is just the sheer extent to which this occurs. So you'll notice that 5 to 20% of all the T cells in the body can become activated and proliferate in response to a superantigen. However, this is really just a massive distraction of the immune system because only a very small percentage will actually have any activity against the infectious agent. So it dilutes the immune response. The immune response thinks it's doing something, but even worse, all those activated T cells are now going to be producing and releasing pro-inflammatory cytokines and chemokines. And you don't have to know the details of this illustration, but it just kind of, again, it's another way of showing this nonspecific activation of T cells by interaction with an antigen-presenting cell. Here's the superantigen, and all these different types of chemical messengers, which in small amounts is helpful, but in large amounts causes damage to the body. So this release, massive release of cytokines, is called sometimes a cytokine storm, and adverse effects include fever, headache, nausea, vomiting, diarrhea, and increased permeability of the endothelium results in leakage of the contents of blood and fluids into the tissues, resulting in a drop of blood pressure. So shock and death can eventually result. This concept of the body actually causing damage to itself by an overreaction of the immune system is sometimes referred to as immunopathology. And you can kind of think of it as a type of friendly fire, again, where the immune system just kind of freaks out and ends up doing much more damage to its own body than the invading pathogen does. 19.1 in your text summarizes the list of a number of virulence factors possessed by Staph aureus, most of which are absent in Staph epidermidis, and this difference likely explaining the differential virulence or pathogenicity of these two species. Some of these we've already discussed. Coagulase, which forms the clots to prevent access of immune cells to Staph aureus. Staphylokinase, which can later dissolve fibrin clots to allow dissemination of Staph aureus through the body. Lipase is produced by both, and this is an enzyme which allows them to degrade lipids, such as sebum, which are present in the skin. And most strains, in fact nearly all strains of Staph aureus, also produce the enzyme beta-lactamase, which breaks down penicillin and allows them to be resistant to that antibiotic. Both have mechanisms to inhibit phagocytosis, although Staph aureus also has this particular protein referred to as protein A, but that, in short, just prevents immunoglobulins and complement from stimulating phagocytosis like they normally would. So those interfere with phagocytosis, whereas other toxins actually lyse cells or create holes, such as this one, this epidermal cell differentiation inhibitor, actually creates a loss of the barrier function of the blood vessels and allows penetration of Staph aureus in that way. We'll talk about exfoliative toxin in a minute. This is present in some strains, and the toxic shock syndrome toxin is one example of a superantigen, and we'll talk about that in the context of toxic shock syndrome. Table 19.2 in your text summarizes some diseases that are caused by Staph aureus. We've already covered folliculitis and the associated styes, furuncles and carbuncles. We'll continue with scalded skin syndrome and impetigo, and in later chapters cover some of these other ones, including toxic shock, bacteremia, presence in the blood, infection of the heart, lungs, and staphylococcal food poisoning. So staphylococcal scalded skin syndrome results from an exfoliative toxin that's produced by about 5% of Staph aureus strains. Young children and immunocompromised individuals are most often affected, and the body-wide rash is a result really of the fact that the toxins that are produced are disseminated in the bloodstream. The skin becomes red, wrinkled, and blistered, and it looks horrible, but fortunately within about a week to 10 days. It resolves, and typically without scarring, because the dermal layer directly underlying the epidermis is unaffected. But secondary infections can occur, and because of damage to the normal first line of defense here of the skin, and those can be due to Pseudomonas aeruginosa, for example, and that can cause some serious complications. So the next few things or conditions can involve Staph aureus or even Strep pyogenes, Streptococcus pyogenes, or sometimes a combination. So this first condition here, impetigo, shown on the left, commonly affects children and results in these pus-filled patches on the face and limb. Impetigo is sometimes called pyo for the pus, derma for skin. Strep pyogenes may also cause impetigo, but again, most commonly Staph aureus. Erysipelas occurs when impetigo spreads to the lymph nodes and then causes a more generalized rash, in this case shown here on the face of an older individual. So this results in again, a rash, fever, pain, and chills. In this case, though, if erysipelas develops, the infection usually results from activity or progression of strep pyogenes. It's important to treat these conditions because both can progress. Erysipelas also has a mortality rate which ranges from 2 to 17%. The causative agent, the strep pyogenes, can also cause a kidney infection if the bacteria get into the bloodstream. The causative agents of both these diseases are typically transmitted person to person or by fomites. Think about a question. Why can we treat erysipelas with penicillin but not impetigo? And this again has to do with which organism is primarily associated with which condition. Most cases of impetigo are caused by Staph aureus, and most cases of erysipelas that involve strep pyogenes. So let's consider some other diseases, streptococcal diseases. While most staphylococcal diseases we've seen involve the causative agent Staphylococcus aureus, in contrast, most streptococcal diseases involve Streptococcus pyogenes. This is also called group A strep, and I think we covered this in a little bit of detail in a previous chapter. But these bacteria can be common inhabitants of the upper respiratory tract, and so in that case, in the same general area as commensal, those people that carry pathogenic Staph aureus commensally. By the way, this term pathobiont means an organism which is established, is colonized, but is not causing disease for the moment, but then can be capable of causing disease under the appropriate conditions. So infections then can occur if the host defenses are weakened, or if some other precipitating event, such as a virulent strain, is introduced. The streptococci can be differentiated from streptococci by cellular arrangements. The streptococci normally growing as chains, in contrast to the clusters seen in staph, but both can produce toxins that lyse red blood cells, and both can be observed to be beta hemolytic on blood agar. You're wondering why strep pyogenes is called group A strep, or you may have also heard of group B strep. So I pulled this slide from another lecture and of course I've taught, just to explain that in a little bit more detail. So the basis of this type of grouping was actually developed by a woman named Rebecca Lansfield. She's shown up there in the upper right of your slide. And she based this grouping based upon the respective carbohydrates present on the surface of the various strep and Enterococcus species, which is a related genus that she studied. And you'll notice that good strep pyogenes because it has this group antigen A, is referred to as group A strep. These are beta hemolytic, and it gives you some of the associated diseases. Group B strep, also known by the species name Strep galactiae, is a concern to pregnant women who may carry this as a commensal, as this can cause neonatal sepsis, so blood infection of newborns, in addition to childbirth fever, puerperal fever, and other infections. And some of these kind of lesser known groups. It's interesting though that Strep pneumoniae and the viridans group actually lack a typable antigen, so these are not classified as a group anything strep. Instead, strep pneumonia is often just referred to as the pneumococcus, and mutans, strep mutans, which we use in the lab, is a member of what's generally referred to as the viridans group. Associated with some diseases of the heart or cavities in the mouth. So let's consider the group A strep in a little bit more in detail. Like Staph aureus, group A strep contains or expresses many virulence factors which make it an important pathogen. What's interesting though about the group A strep is that many of these factors and toxins and enzymes are actually encoded by a lysogen. So if you recall when we talked about bacteriophage and the lysogenic lifestyle, that occasionally a infecting virus of bacterium called a phage can insert its genome into the bacterial chromosome and remain there for many generations, replicating as part of the chromosome during normal cell division. In fact, over 10% of the genome of group A strep is phage. And this is another interesting fact, that there's a chemical which is produced in the human upper respiratory tract, which can actually induce the lytic phase, that is, cause the bacteriophage genome to excise itself and then direct the formation of other phages. The consequence then is that some of these genes can be then contributed or spread to other bacteria by horizontal gene transfer. And you'll remember that that mechanism involving viruses and horizontal gene transfer is called transduction. So again, there's something in our own bodies which can actually induce the exit of this phage genome from lysogeny. Other toxins that are involved in group A strep diseases include rash-causing toxins, and those are called pyrogenic. Don't confuse that with pyogenic. Pyo would mean pus, pyro meaning more a fever-inducing toxin, also responsible for the rash. Streptolysin, similar to enzymes produced by Staph aureus, can lyse many different types of host cells, including phagocytes, so that gives them an extra layer of protection against the immune system. Group A strep, as we've pointed out, are also beta hemolytic. And like Staph aureus, many produce superantigens. In the case of group A strep, one example is exotoxin A. Some of these other virulence factors you'll remember from our previous discussion, but these include capsule. So strep pneumonia, a famous producer of a capsule. And it's interesting that it's made of hyaluronic acid. Again, this is that chemical that connects cells in the human body and kind of probably is one way in which it evades recognition and engulfment of phagocytes. The enzyme, hyaluronidase, and I guess this should have been included also as a list of virulence factors of Staph aureus. In any case, this is again something that can break down this connection between cells, allowing the distribution, dissemination throughout the body. Streptokinase, similar to staphylokinase, dissolves fibrin clots, then releasing them to be able to travel to other sites in the body and establish new sites of infection. And similar to Staph aureus, produces a DNase. that breaks down DNA from release cells and also helps dissolve these neutrophil extracellular traps that we discussed in a previous lecture.